What I find sometimes is some students think that they actually can't do field work, or they're worried about it, and I think they just need a taste of it.
All over the world, 365 days a year, there are scientists working in beautiful places like islands far out to sea, tropical rainforests, and the Arctic. Greenland, the land of tundra and ice, the world's largest island, a place of spectacular beauty and solitude, where the summer sun never sets, and in the winter, it never rises. A place where field researchers flock to study the mysteries of our world. But why? What are they studying? And what would it be like to be a researcher here? Let's find out. Today, we travel with researchers from the University of Maine to get a glimpse of fieldwork in the Arctic. Welcome to Kangerlussuaq, a township just north of the Arctic Circle, home to Greenland's largest airport. Occupying an old military base, this town is a hub for travelers and researchers. Funneling in from all around the world, Greenland fieldworkers start their adventures here at Kangerlussuaq International Science Support. With so much to see and do, research in Greenland is unlike anything else. To get a taste, let's join a research team as they explore the mysteries of the Arctic. Meet Dr. Jasmine Saros of the University of Maine Climate Change Institute. She's here in Greenland studying life in lakes.
I would say I'm a lake ecologist, and that, though, spans a couple different areas. So I look at the ecology of lakes both today and also in the past. Usually when you think about life in lakes, people often think of fish, and they're certainly a major part of the biota, or the organisms in lake ecosystems. But there's a whole suite of organisms that live in lakes. So you can see, starting from the smallest, we have things like bacteria, and then also algae, which come in all sorts of shapes and forms, and they have all sorts of different environmental requirements. And then there's zooplankton, which are the small planktonic animals swimming around in the system. Diatoms are a type of algae, and they're single-celled organisms. And what sets them apart from most other algae is the fact that they have a silica cell wall. So it's almost like having a glass cell wall. So you have a cell inside of a glass box. And we look at them for a couple different reasons. One is that they are very sensitive to environmental change. So again, if you see a change in pH, you might see a certain species become more abundant and other species become less abundant. But because they have this glass cell wall, they leave a fossil behind in sediments. Because if you think about, if you throw away glass to a landfill, for example, it takes a long time for that glass to degrade. And a diatom is kind of the same thing. A sediment record forms very slowly, and you have particulate material, so just basically suspended material in the water column that, much of it will actually be, if it's plankton, it will be consumed on the way down, but some of it will make it down to the bottom of the lake. And so over time you have this material just sort of raining out from the water column down into the sediments, and it forms these layers over time. And so what you can do then is take a sediment corer, which is essentially a tube. You take a core through the sediments, and when you pull the core up, the surface of the core would be the newest sediments, and then you're basically just traveling back through time as you move down the core. In a larger sense, we're looking at the implications of climate change for ecosystems. And so for the organisms in those ecosystems, and how does climate change affect the biology of ecosystems? And in particular, we're looking at lake ecosystems. Over there, then tip of that trail, or we could skirt around the lake.
To study the ecology of Arctic lakes, Dr. Saros and her team travel for miles across the barren tundra. Many times, these journeys are taken on foot, hiking with large bags of gear. To more remote locations, the team travels in style. Once at the lakes, the fun begins. Rubber boats are pulled from bags, sensitive equipment is constructed, and the team embarks to collect water samples and data. The samples are taken back to the laboratory for preservation, analysis, and experiments.
We let these grow for about a week, so it's just lake water with different diatoms in it. And we put them in the growth chamber under nice warm conditions and nice high light, unless of course the mesh is blocking it. And then we let the diatoms grow for about a week, and then we preserve it with Lugol's iodine, which you'll see in a minute, and that preserves the cells. And then we can take it back and count them and see how do they respond to the manipulation.
By studying the mysteries of diatom populations, Dr. Saros and her team help to decipher the history of lake life and the future implications of changes. By understanding what different patterns in diatom populations mean, we get better insight into what the lakes are telling us about changes in large, complex Earth systems. With miles to hike home, we see that fieldwork here is challenging, fun, and important. Now it's your turn to dig in and see what you can find out about the mystery of Arctic lakes. What can these waters tell us? Use the site to collect data, compare, contrast, and ask questions. These mysteries are real. This data is real. What secrets can you unlock about the land of tundra and ice? What secrets can you discover about the world?